introduce you to SeeSAR. The most interesting parts for you, obviously, if you have already projects running, this is where you can continue with the project. If you need a little bit of introduction to the functionalities of SeeSAR and how to do the first steps, there's a small SeeSAR tour which you can click here, and then everything will be nicely explained to you, which you can browse through one by one, explaining what you can achieve and how you can achieve things. And if you don't want to continue this, you can just close the window here. But most importantly for us is obviously starting a new project. This is the interface of SeeSAR, and obviously, since we are trying to do some drug discovery on a target, we need a target. For this, we can load our structure with the menu here, and then can select one of those proteins we have worked with so far, or we can load a previous project that was saved with SeeSAR. The most convenient way to actually get to your target is to use the search box here, which is coupled to the PDB database, to the protein database. And here you can type the PDB code, like for NTJ, and then you will see the target you would like to work with. Or you can use one of the keywords of the title of the PDB, like let's go with maybe something like give it a nip. And then we will see give it a nip. Where you can see an array of possibilities where give it a nip is mentioned. Another new addition to the whole thing, we can also search by chemical synonyms. So if we type now, I don't know, silicoxib, we can find silicoxib, the chemical identifier here, click on it, and then we get all PDBs featuring silicoxib in their structure as well. So to dive in, let's load our first structure. I will go back to the PDB code and work with imatinib today. And I decide to go with 2PLO. And by clicking on it, I directly download the structure from the protein database. And here we are. The most important clicks and tools and keyboard tasks for you. With your right mouse click, you can turn the structure. You can zoom out and zoom in with the mouse wheel. And if you zoomed out too much or lost your compound, you can always focus with the space bar. The first thing I would like to highlight here is now we are seeing we have our kinase with imatinib as the co-complex ligand, and obviously the hinge binding site, so no huge surprises here. Using the control, I can also move around in plane with the right mouse click. And the first thing you will see here, if I now click on my ligand, that now the imatinib pops up. And what you will see here is it's a little bit shifted. So the first thing that SeeSAR does, it optimizes the co-complex ligand with our scoring function, because we need to recalculate the H bonds, and for this we apply our height algorithm. And by doing so, we allow smallest changes up to 1 angstrom of the compound to adapt to our algorithm. So SeeSAR always works with one ligand and one binding site. To define a binding site, SeeSAR asks us here to extract a ligand. We define our binding site by clicking here, using imatinib as the ligands to define the binding site. And by doing so, we now can confirm the binding site after clicking here, selected, not selected, now selected, and then applying our definition. Everything within the target's binding site around imatinib is included. These features side chains, backbone, water molecules, co-crystallized ligands, ions, and everything else. Before we dive into each particular functionality, allow me to guide you through the most important features here. You can adjust the sizes of the windows using the left mouse key. At the bottom, we'll talk in a short about the target view control, and I would like to highlight the functionalities in this section. Namely, if you're interested what the latest update of SeeSAR included, you can click on this megaphone here and will be directed to our website featuring the whole changelog with the newest functionalities and also the past ones. If you need any help in regarding operating a mode, you can always click on the help button where you get your SeeSAR assistant with hyperlinks inside, where you can really look up where you need something. It also features additional links to our beginner's guide, tutorial movies, online help, and the first steps of SeeSAR. The system settings here allow you to adjust parameterization. I will go through them quickly one by one. In calculation, you can adjust which kinds of calculation you would like to perform per default. So let's assume you would like to dog poses, but don't want to perform any height optimization because you would run an additional algorithm to rescore them without any additional subsequent adjustments of them. So you can turn it off. This includes also calculating the H-bond network and also clashes and torsions. So sometimes it can also make sense to not calculate everything at the same time, especially I'm working with a lot of ligands, like 10,000s of them. So therefore, this is an option where you can be efficient and time-saving if you don't need those calculations at a particular moment. In HPC, this is where we connect SeeSAR to our high-performance computing platform called HPC. This is where you can type in how you would like to connect and also your login. In the Inspirator mode, we'll talk about that later on, is where you can select the libraries you would like to use for ideation processes. In license, you can see how long your license is active and for which features and modes and components it is still active. This way you can also add in your new license or request a license if yours has run out. Proxy is basically just setting up a proxy server, quite important if you're working in a larger company with where many people need access to SeeSAR. In this section, you can download the latest connector, as I call it, to the RCSP. So basically just updating the API communication between SeeSAR and the protein database. In the README is a lot of important legal stuff. So basically privacy settings and so on. System log is if sometimes mistakes happen and you cannot follow up why a particular docking or rescoring did not work out. So this is where you can collect this information to provide it to our support team. And we also have third-party integrations, namely Stardog by Optibrium and Yasada, which can be adjusted here for their settings. Next up is the appearance of SeeSAR. Usually I don't work with the light version. I go for the dark theme because for me it's just a little bit smoother and easier on the eye. You can lay out the windows in SeeSAR as well. Some people like to operate like this, especially if like a very long screen can be kind of interesting or work into the height. We can also change the background color. So now it was black. We can also go with something lighter or just adjust it to something, I don't know, grayishly like this. And you can adjust label sizes. So let's assume you have a label like this one and you are a little bit, visually impaired. Then you can use the label size to adjust it accordingly, which is also relevant for publications. Another thing we have considered, obviously, the color blindness. So if you would like to work with something where you can easily differentiate between green and red, make it more adaptable, we also support functionality where the height spheres, so our affinity indicators, can be recolored as well. As for the utilities, the focus view always centers your compound in the middle. You can take screenshots for publications. If you go to the poster, you can also adjust the dots, the DPI intensity, so the pixel intensity, 300, the default setting for publications. This is also what we have considered. Again, you can even select the background color to be transparent. What's quite convenient, you can also export what you are viewing at the moment as a 3D export. So basically, for presentation purposes, let's go with SeeSAR 3D. I will now export it. And in the next step, I can simply open up my PowerPoint, add a new slide, and include the model I just saved, which will look like this. Loading, loading, loading. And loading, loading, loading. There she is. And you can see how nicely it looks. So all of the SeeSAR functionalities, namely also the height spheres, are included as well. And you can also adjust the animation, so really augmenting your presentation to a new level. So much for the 3D export. Back to SeeSAR. In the label and measure drop-down menu, you can see what things we actually can consider during the assessment of your poses or ideation process. With L, show label, you can label anything, be it the side chain residue just to see the number and the amino acid. You can also tag the height spheres, which we'll talk about in a minute. And you can also look at torsions of the bonds to see how well they match empirical data. The next thing you can measure is atom distance. Atom distance is something between two atoms ultimately, so we can measure the distance between maybe this ring system and the phenylalanine here, and you can see it's 3.9 angstrom. You can also do it on the ligand itself if you want to check if the bond length match our expectations. You can also measure the angle. For this, we need to select 3 atoms, and then we get also the bond angle. Measure torsion is basically what I've shown you with the label. I'm now selecting this particular torsion, and you can see the torsion between those is 18.3 degrees. And you can also remove everything that you set up with clear or labels. In visualization, we can decide to hide and show the height spheres, indicators for our affinity algorithm, affinity prediction algorithm. We can also turn on and off about the H bonds. We can turn on and off the torsion coloring, the molecular clashes, if there would be any. Seems there is no such case here in this particular area. We can also turn on unoccupied spaces. So the difference between both is like this. To give you an idea, each particular sphere is exactly the place where one fluorine atom would have place. So this is really kind of very helpful if you would like to evaluate if adding a particular meta group or another functionality somewhere has enough space before doing so, or at least to gather ideas of where you can extend your compound towards to. In some cases, the whole protein is not resolved. In this case, it is. Otherwise, you would be also able to hide and show unresolved segments. And target clipping will be part of our next discussion. So let me quickly go through each particular functionality because this is also a very important science aspect of SeeSAR. For the height coloring. Height coloring works like this. This is our scoring algorithm that we use for assessing the estimated affinity of the compound. And it's based on hydration and desolvation. How it works, let's label maybe this particular carbon here. We calculate the affinity gain and the affinity penalty that you have to pay for each atom of the ligand. This also takes into account that we have to strip off the binding site itself. And it's based on the desolvation. So like this carbon atom gains now some interactions because it's no longer exposed to water. It can form some lipophilic interactions. This is we get 1.8 kilojoule per mole for the desolvation of the ligand. Since we don't form any particular hydrophilic interactions, there is no gain here. As for the receptor, we are stripping off a potential interaction partner in this segment here, because we have likely this hydrogen backbone of the target structure. We are stripping one water off of it, or the potential to interact with one water molecule. We have to pair this penalty, obviously, and this is reflected in a penalty of 4.8 kilojoule. Summing those two factors together, we can see that for this particular carbon, it's not a good carbon. It should be something else, obviously. We have to pay 3.0 kilojoule for the interactions as a penalty. Now let's pick something which is better. Let's take a look at maybe this amine here, since it's green. Here we can see desolvation for the ligand, because it's also a hydrophilic one. This would like also to form interactions with water molecules, is quite high, 7 kilojoule. For the receptor, same thing. We have a backbone here. We have not the backbone, but we have the hydroxy group of the threonine here. Again, we are paying 8 kilojoule as penalty. Yet though, since the ligand and the receptor, the target structure are interacting, we are gaining some affinity back, which is higher than compared to the water itself, which results in an affinity gain of 1.5 kilojoule per mole, indicated by the negative here. Also, if you're asking yourself, what are the interactions partner for each particular heavy atom, you can turn on the eye, and you can see here, in this regard, it's only the interaction with the threonine here. And if you have pick something else, like maybe this one, you will see each interaction that is considered for this assessment. People always ask, how is height performing compared to others? It performs, let's be honest, on the same level as any other predictive algorithms when it comes down to scoring. Nevertheless, height performs the best when you consider hydrogen bonds. It's very sensitive, I have to admit, but the hydrogen bond quality predicted by height is extremely precise compared to what I have been working with so far. Regarding the H bonds, what I forgot to mention, the spheres also indicate how well this affinity, or how large this affinity gain or drop or loss is by the end of the day. So very small spheres mean very small gain, 1.9 kilojoule per mole. It's not so much. Whereas compared to a larger sphere like this one, this is where we get 9.1 kilojoule in total. To give you a rule of thumb, 20 kilojoule per mole are 3 orders of magnitude when it comes out to affinity. So divide by this, if you get like, I don't know, something around 6.7 kilojoule, is approximately 10 times more potent than a comparable compound. H bonds, now I turn them on just to comment on them. H bonds are indicated by dashed lines, and the hue of the hydrogen bonds also indicates how strong they are based on geometry predictions. A very dark green means very good geometry, so this is a very good, strong hydrogen bond. Let's see weaker bonds, so where the geometry is not perfect, like the one here between a water molecule and the carboxylic acid here of the side chain. So light green means geometry is so-so. It's not like it's a very questionable hydrogen bond, but it's not one in perfect geometry, which also can help you to assess how well your compound interacts within the binding site from the geometry perspective. Torsion coloring, so now I turn them on. Torsion coloring gives you ideas about how where high energy conformations are present based on statistical assessment. So let's take a look at the red one here. What we can see here is that this particular arrangement is somewhat not preferred, right? So this is not something that we observe that frequently in a vacuum in reality in PDB structures. And if you take a look from a MedChem perspective, you will see the pyridine ring system and the pyrimidine ring system. They are not in one plane. They are crossing each other. For perfect conformation, for a low energy conformation, both should be in one plane due to the aromaticity of both compounds. So therefore, SeeSAR sees that and also provides you with feedback where you should pay attention to, because it gives you an additional parameter that you can use for filtering of compounds to reassess the quality of docking predictions. And if we now take a look at a green one, like here, we can see the amide and the ring system, aromatic ring system, are in one plane, exactly as we would expect it, without any additional substituents, which leads to steric clashes. So we are good here. It's a green one, and you can also see how frequently this conformation is observed in reality as well. Also, if you don't know which arrangement of the torsion is actually considered here, you can again use the eye to highlight the section. Molecular clash is something we haven't seen in this compound, but we can maybe check it back later on if we have any clashes here. Now about the visualization. This is where we have the target view control. Ultimately, we can do two things. We can adjust color and we can adjust the visibility. So if you don't like to see the whole structure itself, we can go to targets and just turn on the visibility for everything, which will turn on the visualization of every single heavy atom of the target, or turn it off, and then we are left with the ligand only. We can also turn on just the chain. We can also work with different residues. If you are interested only in the binding site, so not everything that this structure brings on the table, just the binding site, then we can turn the binding site on here. Only the residues that are considered to be part of the binding site are now presented. So I can turn them on and off. And if I would like to see only those interacting with my ligand, I can use particularly this one, and then all residues forming interactions with my target, with my compound, are then presented, as we can see now. Same goes also for water molecules. We can turn on all water molecules or none. And likely most importantly, for depth assessment, the target's binding site. So we go to surfaces, we turn on the target binding site. For the binding site only, we are presented with something like this. If you would like to see the surface of the whole protein, we can also do that, which will look like this. And then we can see here how the protein looks like. Fun fact, it's also possible to export the binding site surfaces as a 3D model, as I showed you. But since I don't like to see everything in this woolly clothing, I will turn on only the binding site. If you would like to have a little bit more of transparency, you have to go to color and adjust the transparency here. You can also color it as you like on element coloring, red for oxygen, blue for nitrogen, yellow for sulfur. You can also color it based on log P distribution within the binding site, or can use your own individual coloring, like, I don't know, something like this or this. Ultimately, you will notice that if you have a very, cavity-rich binding site, it's also possible to focus a little bit better by using the target clipping functionality, which will then adjust the representation of the binding site a little bit more conveniently. What we can do here is also adjust the clipping plane, so adjust the depth of the binding site, cut off. We can also flip it to adjust it as much as we need it. Sometimes you have to play around a little bit. We can also flip the whole thing in order facing the other side. And here it's also possible to select whatever you would like to cut off. So basically what should not be shown here, which is chains, surface, waters, and so on and so forth. So this was the very basic introduction of how to operate SeeSAR before we've done any drug discovery so far. Let me quickly turn on the residues and then dive into SeeSAR's different modes. The very first mode is a protein mode. So this is where we interact with the protein structure, can load the structure, can align binding sites, look into them, analyze them a little bit more conveniently. You can also use it to look up or explore your binding sites. So let's assume I would like to know where my leucines are, or I'm looking for maybe a cysteine, not cysteine present. Is there maybe a lysine? Yeah, there is one lysine present. And if I use this one to look for the 3-letter code of the amino acids, it's possible. I can also use it to look up particular residue numbers like 289. Now I'm seeing the alanine here, and every time something is highlighted, we see it in 2D and in 3D as well. Just to highlight once again, in 2D and in 3D. You can also browse through the residues. So right now it's the very first one. If I would like to see the second leucine sequence, I can just click through to highlight them, and they're always highlighted in 3D as well. So it's easy for me to really orient myself and follow up. Yeah, this is the protein mode, and this is where everything starts, okay? For the very first thing I would like to do is actually to show you how you can work with the target itself. And if you would like to modify your target, you can right-click on the protein in the protein window and transfer the protein to the protein editor mode. In the protein editor mode, we can do quite fancy things. We can do small modifications on the protein. We can look up additional binding sites, and we can also look up similar binding sites. Sorry, it's a difference. And we can also perform energy minimization. For the full purpose, allow me to show you. Let's pick maybe this aspartate here. So let's assume you would like to do a small modification. Let's say you want to do a mutagenesis study and see if this particular asparagine has any effect on the interactions with your protein or your ligand. Then you can click on the residue you would like to modify and use those features like adding atoms, like carbon, oxygen, nitrogen. You can change the element, which I will do now. I will change this one to a nitrogen to make it asparagine. You can also change the bond type from 1 to 3 triple bonds, sorry. Not extremely useful, I have to admit. It's a quite niche functionality. But if you have a residue where maybe I have a sugar attached to it, this is how you could actually be able to achieve those kind of things, or even producing tracers or whatsoever, just to see if the volume can handle it. You can also work a little bit with rotamers using the rotation button here. And once you have done what you've wanted, I wanted to generate an asparagine here. I did that. You can then export them and then also work with them continuously. Since we are already here, I'm asking myself, well, what effect does my modification has? So I'd like to do an energy minimization. For this, we have a collaboration running with Yasara. So you can use this button, and you can see SeeSAR and Yasara communicate between each other. And you can select a GPU you would like to use for the calculation, and among several potential force fields to do the energy minimization. Amber is quite prominent, I would say. In our cases, we made quite good experiences with Yasara 2 as a force field, and therefore we'll use this one for the one. You can select if you would like the backbone to be flexible or rigid. In this case, I would keep it rigid. Once we have decided for everything, we go back and start the energy minimization run. This will take a few seconds. There we go. Our new energy minimized structure is ready. We can now transfer it back to the protein mode. Again, we have to select the ligand to define the binding site. We do that. And now you can also see small changes of adaptations, smaller changes here, and also how the angle of the carboxaspartate and asparagine has changed a little bit. I would like to highlight a particular feature that is quite important, but for this we need a very dissimilar binding site. So therefore I now delete the energy minimized structure. Again, I will load my original structure into the protein editor because I only have one active target and only one active ligand each time. Right now I see I'm working with this particular number here. What's the protein I'm working with? This is one I've loaded in just a few seconds ago. And now I'm asking myself, okay, I have my binding site. What other structures contain a similar binding site? For this, we can run the Sienna search, where you can adjust how many solutions you would like to have. And I will do that now. I will search. We are accessing the Protein Plus server and browsing through each one of them, looking for topologically similar binding sites. This is quite important. We are not looking at the sequence similarity here, which you would do in every normal search. We are looking at the topology of the binding site, ultimately which compounds have a very similar binding site. This includes conformational changes, this includes active inactive states. So ultimately, this is a safer bet if you're looking for very similar binding sites. So we can see here the new structures that we have mined from the Sienna Plus server that have the similar binding sites. So basically, you can also see magnesium in some of them. And now I'll just select one of them, the first one, add it back to the protein mode. And if I have two proteins with similar binding sites, I will define a binding site again. If you have two proteins with very similar binding sites, we can use this particular button to align the binding sites. Now we have a 3D alignment, as we can see also at the bottom. Now for the binding sites, similar residues are aligned, and dissimilars, or at least the ones in the same place, are also aligned as well, which can help us spot differences in the topology of the binding site. So what we can do now, we can also turn off the visibility of all residues. And what we can do, or turn on, sorry. And what we can do now is using this particular feature, hide components with similar conformations. And then we can increase this particular setting to hide similar residue sidechains. So what happens now is everything that is similar disappears in this section for the second aligned protein. Sorry, for the second aligned protein. And dissimilar residues are still, for the love of God, are still presented. So let's take a look at this particular pair. I right-click on it and focus. And now I can see in this particular section, leucine is replaced by methionine. Same goes for other residues, like here we have a tyrosine, which is replaced by a phenylalanine, which is also a little bit shifted. So now it's better visible. Yeah, so this is now the complex of the protein editor and the protein mode. What you can also use here, you can also save your proteins and export them as individual files. Quite important if you also align them, they will be all in the same orientation for you. And you can also extract all ligands featured here to different other modes as well. One is the ligand from a structure-based perspective and ligand from the ligand-based perspective. Structure-based drug design, and ligand-based drug design. The only thing they're missing on the left column is the binding site. Binding site is quite fancy. This is where we can explore potential new binding sites. Sometimes you don't have a ligand to work with, like if you import the structure from AlphaFold or use your own homology model, or you're exploring for potential binding sites, other potential binding sites somewhere else, which is not your original ligand. For this, you can just adjust this feature, use this feature here to show unoccupied binding sites. And now you can see here, we have to adjust it a little bit. We spotted 3 potential additional binding sites. One is the orthosteric binding site of the kinase, and we have 2 smaller ones in this section. What we are provided with is the number of residues that form this particular binding site, a dock site score, which is an indicator for the druggability of the binding site. So the orthosteric binding site, quite accessible, 0.59. The smaller ones, way more solvent exposed, more difficult to drug, therefore 0.16. Also other additional parameters like number of hydrogen bond donors and acceptors, as well as the size of the surface and its volume. If you are feeling extremely funny, you can also define your binding site manually by going through residues and add them to the binding site one by one. Purple indicates that this is part of the binding site selection. Same goes also for water molecules. You can also strip off all water molecules from the binding site in the protein editor mode, or go through the water molecules you would like to include within the binding site. And it's also possible to conserve water molecules. For this, right-click on the water molecule you would like to conserve, and then select this water molecule as conserve, and then it will become part of the binding site and will not be replaced during your subsequent drug discovery tasks. Usually we keep water molecules if they form high-quality interactions. So if one water molecule in the binding site forms at least 2 high-quality interactions through hydrogen bonds within the binding site, it will become a fixed part of the binding site. Floating water molecules without any interactions can be replaced during docking. Fixed water molecules that form high-quality interactions are considered as part of the binding site, though. Yeah, but since I like the binding site as it is, I will just skip that and reset my binding site, leaving us with the binding site that we started with originally. Quite important as well, if you are using the command line version of our docking algorithm, FlexX, you can also use the binding site mode to export the binding site definition, because this is way easier to set up in 3D with all the graphical user interface, and then just use this as input for your binding site definition for the subsequent docking steps with the command line. Okay. This ends the section about the left part, so the target structure. Now I will delete this one to have a clean overview and show binding site only. So this is where I would like to keep going. We take the compound into the molecule editor mode. So in the molecule editor mode is where you medicinal chemists, you computational chemists can do a manual modification of your ligand. So maybe let me show you some examples. So by clicking an area you would like to modify, you can select now this hydrogen and use the C key of your keyboard for carbon to introduce carbon, and for nitrogen, F-fluorine, and so on and so forth. And you can also see here, maybe even live, now if I produce a methyl, this torsion becomes green. So we do it also on the fly. Everything we do on the fly, be it tautomers, be it protonation, be it torsion assessment. Obviously, if you introduce a methylene group here, both ring systems have to change, because otherwise they would clash. And this is now, for this part, I will turn on molecular clashes. You can see here we get an indicator that even in this scenario, the methyl group would clash in this particular conformation. So this is something you should consider. Let's see if a fluorine is better tolerated, and yes, it is. So it gives you also ideas about what modification can be used to actually rigidify your compound to some degree to induce particular conformations. And if I now introduce the carbon back, I can also use this functionality to rotate and flip stuff. So this is quite interesting to play around with. You can see what problems may arise when you introduce a particular group. So clashes are now displayed, and also with different levels of how much things clash. Red is very bad, orange is a very high energy conformation. So this also helps you to sample the space around a compound of interest. What's also quite interesting, it's also possible to, in the flipping, now I have selected the small compound. So you can see everything on the right side is grayed out. So this is not changed, this is not affected. We can also switch that. Now we are keeping our methylpyrimidine fixed, and now we are working with the rest of the ligand. So basically, if you want to switch sides of whichever residue or moiety you would like to rotate, this is your way to go. Okay, now I have done some bad things, so I can also undo and redo them just to see of what I did. And again, here you're always working with one active molecule, which you can see here. And if you clicked farther away or clicked another compound, SeeSAR will ask you or inform you that the molecule editing is now paused. And if you'd like to continue to resume the whole session, the editing process, you can just click on it, and then you will be back in the place you left SeeSAR at. Here, it's now completely up to you how you would like to do things. You can also introduce ring systems. Right-click on the target, and then let's add a tyrosine here. And I personally asking myself, okay, and now this is a good edit. How would it look like in reality? So right now I will save this compound, and it will be added to the table. We will run an additional height assessment to see how we can salvage the situation. And by doing so, smaller adjustments are allowed. And we can also see here in the traffic light scheme what problems may arise or can exist here. You can see we have intermolecular clashes. So basically the tyrosine now clashing with our pyrimidine here. No intermolecular clashes, so no clashes with the target binding site, but our lipophilic ligand efficiency has dropped a little bit. So this is a traffic light scheme, and when we are doing virtual screenings or assessing results from a docking campaign, we try to consider only those with the best quality, meaning where everything is green only, which is also where height performs the best. Another feature, so let's restart the whole idea. We are starting again with the editing. I click on my compound. I would like to edit this compound. There I go. And now I would like to maybe microcyclosize a little bit. So I'm doing something weird. And I would like to connect this particular compound to this area. So I connect both ring systems, and now I'm using change bond type 1. So I'm cyclizing the whole idea here. And once I'm finished, I would like to use the template stocking. So now I'm not exporting the compound and allowing only one step of optimization. Here, I'm actually telling for the edited compounds to be docked in the same fashion as our query compound, the one we've started with. And let's see what happens next. So I click on it. SeeSAR starts the post generation, calculates the affinity, and there we go. So we can see our compounds have merged quite nicely. And compared to the original compound, if I would like to pin my compound, my original one, I can do it here. I can also recolor it in red just for a second so we can compare it a little bit better. And we can see that our edited molecule was docked perfectly on our initial starting point. It's also possible to adjust the protonation state of compounds. So in this case, we are now looking at this particular residue. Let's assume you know from experiments that it's not this nitrogen is the one which is deprotonated, where we can also change the charge. So now we neutralize it, but it's actually this nitrogen here. Once you've finished, we can export it and see. SeeSAR doesn't allow that because it thinks that this one is still better. But ultimately, in some cases, it can work out if the environment considers that as a valid solution. In this case, obviously, it considers this one to be the real one, which also makes sense given the idea that here it can nicely interact with the backbone, whereas in this case, there is no sophisticated interaction part. For the next miracle, I would like to go into the first docking. I would start with the external docking mode. The external docking mode is our HPC response to visual screening, to docking ultimately. So we thought it doesn't make sense to run huge visual screening campaigns on your own hardware. SeeSAR runs very smoothly on very standard, slim hardware, but nobody runs a visual screening of 5 million compounds on his own home desktop. And therefore, once you have installed an instance of our HPC platform, you can also use this for visual screening purposes. The way it goes is you have your binding site definition, you define your binding site, everything is well set up. And then you can go here, load molecules from your own library. Just, I don't know, let me quickly add 3 random compounds here. And then send them over to your external hardware where the calculations are done on an external server. And once they're finished, then you can just subsequently download them. You can also upload your libraries. So we also have a library management system in our high-performance computing platform. I have uploaded 10 molecules as a library there. I will just select them and start a docking campaign. So now I'm sending all my files to St. Augustine, which is approximately 25 kilometers away from me. And while this calculation is taking place, I will guide you through the original docking mode that runs on your local machine. The original docking mode is exactly what it is, the bread and butter of interaction predictions, of the binding site prediction, of the binding mode prediction. So let me load a library of some compounds to dock. I will now load some molecules. Let's go with something for the docking mode. So here I have a random set of 10 compounds. You can also load any compounds that you like just with smiles, and SeeSAR will automatically generate 3D coordinates for those compounds. And what I would like to do, I would just like to dock my compounds. For this, I have many settings I can utilize. I can use pharmacophore definitions, defining interactions that I would like to see in my structures. Oops. I have to zoom out, sorry. Binding site. So let's assume I would like to have an interaction with the backbone here. So now I'm having this kinase, right? A kinase always has this hinge binding motif. Now I would like to form an interaction with this particular region. I show possible interactions, and now for the perfect geometry, an interaction spot is calculated. I can set my sphere to this particular region. increase its size if required, and select the pharmacophore constraint I would like to have there. I have a huge array of selections from hydrogen bond acceptors, donors, to aromatic residues, halogens. Honestly, I forgot how many pharmacophore constraints we do have inside. What is quite useful, you can also use your own smarts definitions to set a pharmacophore constraint. In this case, I will just set hydrogen bond acceptor in this section. It's also possible to define exclusions. So sometimes you don't want particular features in one region, like you don't want too many rotatable bonds somewhere. So this is how you can also exclude pharmacophores. In this case, I would like to include it. So this is mandatory. And if you have also several of them, you can also decide if they are optional or not and how many of them have to be fulfilled. So basically very sophisticated pharmacophore handling. And now in the settings, you can decide what kinds of, how many poses you would like to generate. You can play around with the clash tolerance. Basically, default is a very normal van der Waals radii. If you have a very tight binding pocket, it could make sense to reduce the van der Waals radii of the heavy atoms of the target, which you can then do by increasing it to medium or high. I don't know who would like to do that, but it's also possible to allow additional ring conformations of high energy, like the boat conformation. By default, only the chair conformation is allowed. Twist is something people can consider if they would like to, but I would recommend that as well. You can also allow the stereo center flipping for R and S, E and Z, and both at the same time. So especially if working with SMILES catalogs that do not include stereochemistry, you can use those to sample all potential conformations of your stereoisomers of your compounds. And also for the flexible covalent attachment, basically if you have a covalent attachment point like at cysteine, you have to imagine it like this. Flexible means that it can rotate around the cysteine. In some cases, we are quite confident about the exit vector of your covalent bond. So basically the cysteine and the hydrogen have to face in the same direction always. It's exactly this direction it has to go to. You can also make it rigid here. If you need pharmacophore definitions for your Flex X command line, you can export all the pharmacophore constraints as well using the export definition here. Let's start the docking, I would say. We offer 3 different docking methods that we can check one by one. The standard docking is exactly how it sounds like. You take your ligand, you throw it at the binding site, and you check for whatever survives. We're generating 10 poses per default, no constraints per default, but you can still apply pharmacophore constraints. In general, in almost every single mode where a ligand is involved, you can apply pharmacophore constraints. Now we're running. The standard docking mode is a mode where the backbone is kept flexible, also the side chains, so the target itself is rigid. Nevertheless, there are all kinds of heavy atom flips, like in a glutamine, histidine, asparagine. This is possible. We do sample those kind of things, so the flipping between nitrogen and oxygen in the amide side chain. We are done, but the overall target binding site is kept rigid. And this is how it looks like. Our samples are there, our results. You can see that our pharmacophore constraints did seem to work. So the interaction is there. And we are also giving informations again about traffic light schemes, where you can see which ones survived and which ones did not. So this is the standard docking. Now for the template-based docking, we need an initial compound to work with. For this case, I will just go back to my protein mode. This is where my imatinib awaits it's time, I will select imatinib and use it as a reference in my docking mode. So now, space docking, sorry, misclick. I will use imatinib and use it as reference in my local docking mode. Now I can see it's set as reference, as a template. Now I do not need the pharmacophore any longer. And now I can start template docking. Template docking from our side is fuzzy. It works a little bit more convenient that it's mere substructure. Also match a phenyl and cyclohexyl. So we need at least 5 heavy atoms to do a matching. It's 10 times faster than the standard docking, the free docking. And what we can see here now, SeeSAR tried its best to align those poses, so the amides and those side chains are now overlapping. So see, it's exactly doing what it's supposed to do from its perspective. It tries to match it in the best possible way, and like in this case, the phenol system until it's fine. So yeah, template docking. And for the last part, for the docking mode, I like to clear it up a little bit. I'll now load 3 ligands. I just copy pasted the smiles for a covalent docking showcasing. So in this case, we now have 3 compounds, each containing a covalent warhead. Here they are. And what I would like to do now, I would like to perform a covalent docking on a residue. So those are tyrosine targeting covalent warheads. And surprisingly, we also have one tyrosine here. We can also see attachment points for covalent inhibition that are by default included in SeeSAR, highlighted in bluish, like with this blue cylinder. And if I'd like to dock them onto the tyrosine here, I right-click on it. Now it's highlighted. This is now my covalent target for the docking. And now I will press the covalent docking button, and we'll get the results within a few seconds. And what we can see here are the results. So estimating covalent binding activity is very different for covalent inhibitors. We all know that. So therefore, we provide an endless tag to the left side just to indicate the covalent inhibition here. And what we can see here, again, the covalent attachment point of the ligand to the cysteine, to the tyrosine here. This is also indicated by an R, and then how a ligand is predicted to bind there. This answers maybe some questions here, or raises the question, how do we do that? So we detect covalent warheads automatically. We have a set of approximately 40 covalent warheads that are automatically recognized by SeeSAR. If we perform a covalent docking, they are then translated into their bound form and then attached subsequently to the target residue. So everything is very convenient. I have never seen a covalent docking that is so straightforward, so sophisticated. So I really love this one. And that's about the docking mode. It seems like I received a message, and the message is that our external docking run has finished, which is lovely. So let's go back to our external docking mode. You can see I can switch between the docking modes here and here by hovering over the docking button. And now in my external docking mode, I am presented with the button to download the results from the server, which I will do. And you can see here also in the external docking mode, all modes are supported: standard, template, and covalent docking. Now I will download all the results back from St. Augustine to my own local computer. And here we are. We have 92 poses available. And I can click through them and see what the results are. So obviously, it takes some time to work with a larger set of compounds. So therefore, we have taken also this into consideration, how we can make it more sophisticated, because here we also include triplings and doublings and so on. We generated 10 poses, and 10 poses are quite a lot. So let's take this structure and see if we find duplicates or different poses for this particular structure. And we do. The second one, we find even a third one. So ultimately, obviously, each medicinal chemist will ask themselves, or each chemist will ask themselves, which one is the best? For this visual assessment and for post-processing of docking results, we have implemented the analyzer mode. I will now transfer all my ligands. So if you want to move a chunk of ligands, you can always click here, check all, and now I take all of them to the analyzer mode. In the analyzer mode, we have options to filter compounds as our results. The most important feature here for you guys, you have to click on this funnel here, is this one, group molecules. If I click on group molecules, I will create groups of my compounds, and only the highest ranking, so the best predicted affinity, will be displayed. It's also possible here to apply additional filters, like for drug likeliness, rule of 5, for fragments, rule of 3. You can also apply different filterings for torsion quality. Let's do that. I want only the good results, only green ones. I also want only those without any clashes, or at least the best ones. You can filter for molecular weight, total polar surface area. So a lot of parameters are already calculated by SeeSAR automatically here. I will dive into that in a second. And once we are finished with setting up our filter parameters, we can click back on Apply Filters. And we see for those random molecules that I have sampled, only a few survived, but all of them have green torsions and green clashes. So very trustworthy poses. In this regard, we allow up to 2 orange unsubsequent torsions for the compound still to be considered as a green torsion compound. More than 2 will result in an orange clash, and subsequently in a similar manner, we also do it for red. It's also possible to filter compounds by defining pharmacophores. So ultimately, we can reactivate the pharmacophore setting we set up in the docking mode as well. And now that the constraint is active, we can filter again and see, okay, only 3 compounds have survived this. You can also use this for smarts, obviously. So again, pharmacophore constraints work almost everywhere in SeeSAR. You can also use it to define pharmacophore constraints to be used as export for your command line applications. And as I've mentioned, some people always ask, I get asked the question quite a lot, how does SeeSAR perform? The good thing is you can check it with your own in-house data. So ultimately, you have your compounds, you have your activities, and with the height performance check, which is placed here, you can couple the affinity prediction of height to your biological data to see what the R-squared values for your compounds are, so the correlation. Yeah, so the analyzer is a very sophisticated tool. What people miss every now and then, you can expand the table here. So basically what you would like to see, you can increase the parameters you can look out for, like the number of rings. You can also, I'll talk about it in a second, rotate number of rotatable bonds. So also if you import SD files into SeeSAR, this is where you can also open them up and then reuse them again as your filters in SeeSAR. So basically how to adjust the table and also how to filter compounds here. Some button is always overseen. namely the grid view. So you can switch from table to grid, and in the grid you get those fishnet diagrams where you can adjust them. Oops, sorry. Where you can adjust them. So you hover over the fishnet diagram and going to the gear, you can set up new properties you would like to have, like, I don't know, number of stereo centers. I allow only one, otherwise it will be a bad compound. I like only two stereo bonds, and basically you get then a nice visual assessment of parameters that are of interest to you. So also again, for a quick check, subsequent post-processing, nice to play around with. What I haven't mentioned yet is our second third-party implementation, namely Optibium. If you have an Optibium license or would like to use Optibium within SeeSAR, it's just a nice add-on price-wise. You can also calculate the properties. I will check all compounds, calculate Optibium properties, and then what I can get are ADME properties that are based on the Optibium prediction that can be used again for further filtering and assessing your results. So basically blood-brain barrier penetration, PGP category, their predicted log P values, their predicted log S values, the protein binding fraction, and so on and so forth. So also valuable tool if you have Optibium on hand. It's quite nice, but it's also possible to just not purchase the whole Optibium package, but only this particular model to play and SeeSAR around with. And what's even more important, it's also possible to use those molecules as input in Optibium's StarDrop software. So both platforms can cross-communicate to a certain degree. It's also possible, obviously, to save your compounds. So if you have marked all your compounds and would like to use them in another tool for cross-docking, I don't know, for MD simulations, you can export the molecules as molecules only, so only the molecules with the 3D coordinates as well, and also all the calculated properties that are listed in the table as well. Or you can also export them as complexes. For export as molecules is possible as SD file, and complexes supports MMCIF, as far as I recall. Welcome to the Similarity Scanner. The Similarity Scanner is a ligand-based mode where we align compounds without the need of a target structure. I have prepared an example, and this is the Similarity Scanner. So let's assume you have your imatinib as a target or as an active compound, but you don't know the target, and would like to know how do the other compounds actually align to my initial compound. So the first thing we have to do, we have to define a template molecule to perform 3D superpositioning of the subsequent compound. For this, we right-click on our compound, use this as a reference in the Similarity Scanner. Now we see the template molecule is loaded. We have the same feature variety as other parameterization options as we do have them in the docking mode. We can define pharmacophores. We can define the number of poses we'd like to generate. We can also adjust the minimum achievable score. So basically anything lower than 0.6 becomes quite chunky. So we don't even calculate this or do not display them. And again, we are allowed to perform additional selections of the ring conformations. The Similarity Scanner also exists as a command line tool called FlexS. This is the underlying algorithm. So if you need also the pharmaco definitions for the command line tool, this is where you can export them. Since we have set up everything, more or less, we can just initiate our run by pressing on the Similarity Scanner run button. We should it's also because it takes up CPU capacities. So we are informed that 44 compounds were not able to survive the whole process, and you have to consider it was less than a minute for all of those compounds, and 101 compounds were sampled. For the rest 44 compounds, the score was just too low, so therefore they are not considered to be alignable. The rest survived though, and this is how you can see the compounds here. You get your reference compound, our reference compound, and the alignment with the result molecules. You can also rank them. Let's see what's on top. So on top you have the original compound, which is no surprise, I would say. And we have also smaller derivatives. And the way you have to think about the similarity scanner tries to match pharmacophore features of the compounds. So it's not only focused on shape only. Let me see if I can find maybe a nice example. I think this is somewhat closer. Yeah. So ultimately what you can see here in this section, you have to imagine this nitrogen is considered as to be not protonated, so it's a hydrogen bond acceptor in this particular case. So therefore, this amide, carbonyl, oxygen is considered to be matching because it's also a hydrogen bond acceptor. As for the nitrogen here, this one is protonated, and therefore this particular region is aligned with the hydrogen bond donor in this case. I also saw another example with this one. So here you can see, here we try to match the lipophilicity of this methyl pyrrole to the phenyl ring system, just to capture the lipophilicity pharmacophoric feature of this arrangement. Unfortunately, it's not possible to really group those molecules by the best candidate because the way it is constructed now, we are working on that currently. Grouping takes only height calculations, so the affinity calculation into consideration. Nevertheless, it is possible to export all of those compounds. You will also receive a numeric value for each entry here. And then working with the SD file, running some Python scripts. You can also reach out to me. I have a Python script for that, to mine for the best aligned candidate for each individual candidate of those compounds as well. So what is the similarity scanner used for? Ultimately, it is considered to be used as a virtual screening engine if no target structure is available, since you're aligning compounds on an active compound that you have on hand. This is the chemical space locking mode. We already have a lot of materials available on that. Therefore, I will keep it very brief. This is a structure-based approach where we work with our ultra-large chemical spaces to mine for the best possible candidates. Out of them, I will use now our WOC1 test space. So basically, this is a sample space just to see if the setting works for you or not. The first thing you have to do, you select the chemical space. Right now, I have only 2 of them. I will select the WOC1 test space. Then I can start the chemical space locking engine. So basically, I have the same possibilities as I always do. I have the possibility to select pharmacophore constraints. I can select docking parameters. By default, we only generate 5 poses for the syntons in the first step. And then let's go. So the results are now sent to St. Augustine. I can also check on the progress here. In the first step, the syntons, so basically translated building blocks where the functionalities are encoded into extension vectors, are now processed, are now docked into the binding site. And during this period, also the chemical space is ramped up. The whole test space consists of 30 fragments, basically a three-component reaction where one compound is attached to a middle one and then ending with a final tail. So basically 10 times 10 times 10. We are now browsing through 1,000 compounds currently. If I would have more spaces on the server right now, I would also see all the available spaces that can be used. Right now we have 7 providers for chemical spaces that you can use for ideation purposes and also to purchase those compounds. And you will also find all the details like the web page and orders once the chemical space is uploaded to the server. So we are already done. Let's check the results. Now for the steps, everything is sorted by ligand efficiency per default, height-based. I will now zoom in. So this is the first fragment, looks good to me. And if I would like to use it, I will click on it and use it as input for my next step. So, but now I have to select the best candidates I would like to use for follow-up. This one may be even reasonable as well, why not? I would just pick a random of 4 compounds, 4 fragment symptoms, the starting points. This is something we already have. This is also something we already have, I guess. Maybe this one just for the funsies. So now you have selected the compounds. We usually recommend to go with at least 100 compounds, symptoms as starting points. And now we are moving those that we have selected into the next extension step. In the next extension step, we are looking at all potential compounds that can be synthesized using our initial fragments as starting points. So we don't waste time to look into bad candidates, right? If you have a very bad starting fragment, it doesn't make sense to follow up with, doesn't make sense to consider everything around it for those kind of purposes. We are already there. Extension 1. Now I want to view the results. I'm downloading them. And here we are. So now we have already 119 compounds that we were able to document the binding site. We can see the first fragment was now extended. I would also like to continue with that. So here I'm feeling like, okay, this could work, but there's a clash. I don't want to follow up with those compounds. This one looks also nice. Let's take this one. It's also possible to rank those compounds based on affinity. I wouldn't recommend to do it for the fragments though, because then you have a bias, you're working with larger fragments in the chemical space talking based on our experiences. But this one may be funsies. Always pay attention to picking only the green ones. So what about this one? Sure, why not? So now I have 3 compounds selected. Again, it's just an example. Since I would like to finish them, I will now take them to the second extension step, which is officially step 3. Now we wait for a very brief moment. Now basically again, 3 times the final compounds, 30 compounds are now generated with 3D and docked, template-based. Let's check the results. And here we are. So these are the final compounds. I can also show them in 2D. And now it's the same as for every visual screening. You go through them and pick up the best candidates to follow up with. You can also guide everything using pharmacophore constraints. Again, everything is covered in our more elaborated tutorials because it's a very large topic, so I don't want to waste too much time here. But chemical space docking is quite powerful. It's also possible to add the top molecules to the results table. So you can also download a large amount of molecules in one go. Usually you download only how many? Up to 50 or 100 compounds in one session, but you can also go up for a larger amount using this particular functionality here. This was about the chemical spacehacking mode. Now I would like to go into the Inspirator mode. For the Inspirator mode, I will take my initial compound and transfer it to the Inspirator mode. And I will quickly guide you through all the functionalities that we have. The very first functionality I would like to talk about is the analoguing. Analoguing performs smaller modifications on your compound, something that an everyday chemist would do on a daily basis, checking for smaller adaptations of the structure. Approximately 260 transformations are encoded into this particular feature. And if I click on it, you will see the name of the compound has changed. This explains what it did. It exchanged the phenyl moiety for quinacylone. You can see it here. The modification is also highlighted. And it goes on explaining what it did. So basically, those are smaller modifications up to the size of adding a phenyl ring system. So a maximum of 6 additional carbon atoms. Also taking bioisosteres into consideration, adding amines, hydroxy groups, and so on and so forth. Sampling which modifications you would like to have. Again, you can define pharmacophore constraints throughout the whole Inspirator mode and also adjust how many solutions you would like to have per session. Now I would like to get rid of the analoguing results and show you more of the other features. The first thing I would like to show is the grow algorithm, fast grow. Now I have to resume with my target structure. By clicking on a bond in 2D or 3D, I can decide which part of the compound I would like to replace. So let's assume I would like to go from here. What happens now, I will keep everything that is still in cream, beige, whatever, sand colored, will remain the same. Everything that is grayed out will now be replaced. This is how also you can set up the direction of the transformation. We see fast grow is glowing, and what happens if we click on it, we'll now sample the topological parameters and properties of the binding site and look for the best fragments to complement the binding site. Fast grow is called fast grow because it is extremely fast. Right now we are browsing through 120,000 fragments with 10 conformations each within seconds, looking for those that can actually expand into the binding site. And this is where you get ideas for modifications. You can see here very MetChem-like results, I would like to say. Yeah, the add side maybe not so much, but still can be considered. And the best thing is, if you're not satisfied with the results, you can request simply more. You can also adjust within the settings with the parameters to get more results per default. Let's get maybe another 10. Maybe this time in this direction. And there we go. More results. We also see very nice decorations, also complementing intramolecular H bonds. And again, if you're not satisfied with the results, you can always guide your searches by applying pharmacophore constraints. This is FastScroll. Again, I will quickly get rid of the results here. And continue with the Inspirator. Quite important, FastScroll comes in different colors. We do have a huge variety of databases dedicated for particular functionalities, peptides design, hinge binder motifs, medchem-like libraries, sp3 rich decorations. So ultimately, we do have different varieties of sets to use in FastScroll with, depending on your use case and what you would like to achieve. So basically, a very tailored solution to enhance the performance of the tool if you're working with a particular target. Next one is recall. I'm doing the same thing. I would like to replace sections of my molecule. This time I'm applying 2 vectors, meaning I will keep this particular tail of my imatinib and the pyridine here and replace everything in between. Recall works as a 3D replacement tool. It also works in a vacuum, so you don't need a target structure. You can just load the 3D coordinates of your ligand and then apply recall. Quite interesting if you have a very nice pose, maybe of a PROTAC, which is energy minimized, and you're asking yourself, okay, what is something that you can use as an exchange for this boring linker here, or just sample more linkers in this section. So not completely reliant on structures, and it's a very 3D coordinated mode. So now we can see here, we're getting proposed a pyridine and a Q chain. I don't like that much because it's so flexible, but we get very reasonable ideas how to replace this particular section, very straightforward solutions as well. Also some of those that are not so completely interesting, maybe because too flexible. In those cases, I would like maybe to go back and say, okay, let's apply pharmacophore constraint in this section here. So I would like to have here an aromatic system, so no rotatable bonds. I'll do another 10. And you can see also the pharmacophore colors are actually different based on whatever constraint you applied. Green for acceptors, red for aromatic, and so on. So now to the new stuff, and you can see, okay, now we got the ring system back. Ring system, ring system, ring system, ring system. So yeah, this is how you can also guide recall. A smaller thing that people usually struggle with in fragment-based drug design, FastScroll can also be used in fragment-based drug discovery. So let's take our imatinib and maybe split it up a little bit. So now I would like to keep maybe only those features. So I've split my imatinib into 2 smaller fragment-like compounds. So this one, now I take this one to the Inspirator. I will turn off the constraint, turn off the constraint. Now I'd like to go from this hydrogen here. So I select the hydrogen, again, FastScroll. Let's see what we can find. We are browsing through 1.2 million conformations in this time frame, just for everybody to be on the same page. And we got some ideas where we can grow those compounds. Obviously, the results here are not so large because they're quite small. The binding site is well defined here, so this may lead to clashes here. But even in those cases, you can do an iterative approach. So let's see if we can find something like this. We can take this one. We use this as a starting point and then keep on going with FastScroll. Well, things went a little bit downhill, but at least you get the idea of what can be achieved with FastGo in this manner. Another scenario for fragment-based drug design is compound merging, fragment merging and fragment linking. So taking back to the molecule editor, we have our two smaller fragments that we have created. And what I will do now, I will transfer both at the same time into the Inspirator mode. And now I will try to connect them by defining exit vectors on both of them. I don't know if they were too far off. I didn't try this in the first one, so let's see what happens. I'm using the same algorithm as we did for the scaffold replacement recall. Now it's linking and merging. Yeah, it worked. And here you can see ideas how to connect both compounds in the best possible way. So this is very stupid, and this shouldn't happen. I'm actually quite surprised. This is a mistake in the algorithm. But there are some alternatives, at least accordingly. So they are quite far off. This is something we all agree upon, but there's also more data. So recall ultimately, again, you can apply pharmacophore constraints to look for only ring systems to make that widget. What's also quite important, we do have also several recall indices available, so several recall databases. Again, very oriented based on the PDB itself, based on crystallographic data, based on ring systems. So basically a lot of database you can work with to really come up with the best solutions for your project. This was the last mode I see in SeeSAR, which leaves us with a possibility where you can ask your questions.